we move over to, thanks to Czechia once again. We move over to another corner of Europe. We're closing in on Italy, and we have Riccardo Risso and Raman Parasuraman, who comes from the Advanced Light Microscopy Italian node. And they come in with an exciting lecture on Golgi maturation-dependent glycoenzyme recycling controls, glycosphingolipid biosynthesis, and cell growth via GOLF3. Raman.
Good. So before going into the talk of Riccardo, which will be more about cancer, I thought I'll give you a brief presentation of our node. As you know, we are located in Italy, and we are composed of 5 centers spread all over Italy. And I've been talking from the Naples center down south. And then each of these centers obviously have we all have all the normal technologies, silicon, focal, LEM, everything. And plus, we are also specializing. Each node has its own specialty. For instance, our node, we are developing more correlative light electron microscopy methods and also FRET imaging, while Genova is an important node for super resolution microscopy. And each node has its own specialty. Each center, I mean, more than node. And we, other than that, each of us also have our own applications. We are also not only we offer services that we use. We provide microscopy things that we use. So in the Naples, for instance, we are concentrating on cell biology and membrane trafficking and signaling, also cancer biology. So most of the techniques that we offer to the users are something that we will be needing ourselves. And other than that, we also develop methods. Each center also has its own method developmental unit. Well, Genova, for instance, they develop a lot of volumetric imaging methods and super resolution microscopy. Naples develops methods about CLEM and also FRET-based imaging methods. And we again develop probes for super resolution microscopy. Padova is good in functional imaging. They develop probes for to visualize calcium spikes in the brain, while Florence again is developing techniques in functional imaging. And Milan is a very huge facility with developing methods in high throughput and other things. I wouldn't talk about the common techniques that we use, and also probably Riccardo will present a lot of them. I'll just concentrate on 2, 3 that he won't present. Okay, and this focuses on Naples, but similar things are also available in other centers in the node. One is we provide tomography of, this was a Golgi apparatus. And then we also do FIPSM imaging to see large volume imaging. Plus, oops, it's not moving. And we, as I told you, we were one of the first in developing the modern CLEM. It was almost 20 years ago. We use CLEM imaging to see on adhered cells to see structures moving in coupling video microscopy to electron microscopy. And we also, as I told you, we develop probes for super resolution. Here is one of the probes that we developed for storm imaging. Using this probe, which was regioselectively labeled FAP fragments, we were able to actually visualize microtubules almost to the native microtubules, almost to the same structure that they represent. So I leave it here, and then I think I'll ask Riccardo to take over to say exactly what he did using the facilities in this microscope.
So thanks, Raman, and thank you all the organizer for giving me the chance to share with all of you the data. So I will start the presentation from a basic concept that is the cell sociology. You know, cells in our body are not single entity, but they are part of a community. So they interact between themselves and the extracellular environment, thus experience social life. So they communicate, exchange material, but when this communication goes wrong, then disease happens. For example, cancer, in the context of cell sociology, can be considered as a social dysfunction within the cellular community. This communication anyway happens not only between cells, but also at the intracellular level. For example, organelles in eukaryotic cells exchange material, protein and lipid in order to communicate. To do so, they eukaryotic cells develop different strategy of transport in order to exchange, information from a donor organelle to an acceptor organelle. And here you can see some example, okay? So two organelles can communicate by using small vesicle or secretory granules or intercisterna connection, tubules, and many other models that you can see here. And all these strategy of transport are actually used in this endomembrane system, extensively using this endomembrane system that is the secretory pathway. Okay, so the secretory pathway is extremely important because 1/3 of the human proteome use this endomembrane system to synthesize, transport, modify protein and lipid before delivering them to their final destination. But let's see how it works. So proteins are synthesized at the level of the ER, and after folding and quality control, they leave the ER and reach the Golgi. This central organelle of the secretory pathway mainly does two things. So transport proteins from the early to the late Golgi. And during this transport, proteins are decorated with sugar moiety, a process that is called glycosylation, before being delivered at the TDN level to their final destination, the endolysosomal system or the plasma membrane. So Golgi, as I was saying, mainly exert this process that is called glycosylation. So this is exerted by glycosyl transferases and glycosidases that are enzymes which are distributed from the early to the late Golgi in a specific intra-Golgi sublocalization in order to guarantee an appropriate glycosylation. So glycosylation is important for many reasons. So it works, sugar affects solubility of protein and lipids, but also represent targets. Target for the entry within the cell of bacteria, virus. And so it's extremely important. And what I'm going to talk about today, I'm going to talk about a molecular machinery which regulates in the Golgi the localization of Golgi glycosyl transferases. A machinery that when it's impaired, it causes proliferative diseases. But let's go a little bit more in details. So the Golgi mammalian cells is organized like this in this cartoon. It's organized in a ribbon-like structure that under confocal microscopy appear in this way. So you have a cis marker in green and trans marker in red, which overlap mostly 90%. But there are tricks which fragment the Golgi in functional Golgi mini stack, which are this spot. But then if you zoom, then you can see a nice separation between cis and trans site of the Golgi, cis in green and trans in red. Okay, so you can use this assay to study the localization of intra-Golgi, Golgi enzyme or protein that are crossing the organelles. And you can study using electron microscopy the morphology as well as the localization of certain Golgi enzymes. This just to tell you that all this strategy, all this microscopy technique as well as assay has been used to answer one biological question that is related to the mechanism of intra-Golgi transport, which appear in Wikipedia still as an unresolved problem in cell biology. I will show you directly which was the model that came out after several studies. And what we find is actually that, as you can see from this animation, proteins, once reach the first cis cisterna of the Golgi, they remain stuck there. And the cisterna move forward in order to allow the transport of the protein from the early to the late Golgi, a process that is called cisternal maturation. During this process, Golgi enzyme, here represented in different color, they recycle back using this COP1 coated vesicle in order to maintain the steady state distribution. How we reached to this model? We just took cis Golgi enzyme in green, here mannose 1. We blocked the recycling of this enzyme. We actually find that the enzyme leave the original position, the cis Golgi, and move to the late Golgi once we block the recycling. Then, once the enzyme reaches the trans site, if we allow it again to recycle back, and move to the original position. Okay, this was the first demonstration in mammalian cells that Golgi enzyme recycled. But which is the molecular mechanism behind this process? To go deep in detail in the molecular mechanism, we study in the literature and we find out that there is a protein that in human it's called Golf3, which works as an adapter. It binds Golgi enzyme at the level of the cytosolic tail. In yeast, Golf3, it's called BPS74, and has been found to be a component of the recycling machinery, meaning that Golf3 in yeast binds the enzyme at the level of the cytosolic tail, insert them in this retrograde COP1 vesicle, and by doing so, it allows the retention of the enzyme within the Golgi via a recycling mechanism. Why this protein is important? Because apart being an adaptor, is also an oncogene, okay? It's an oncogene because when it's amplified, it's overexpressed, and this overexpression causes the activation of mTOR, and which in turn causes growth and proliferation. So what we have done as a first experiment is to identify most of the Golgi enzyme which interact with Golf3, with this adapter. To do this, we have used this assay. We have taken avidin beads, we functionalized them with the cytosolic tail of Golgi enzyme, and we use these beads as a bait to pull down Golf3 from total cell lysate or purified protein. Using this tool, we identify most of the enzyme which are clients of Golf3. And the interesting thing is that these clients, they are part of a specific pathway, that is the glycosphingolipid, the biosynthetic pathway, which I'm going to talk soon. Anyway, the next question, once we find the interactors, I mean the clients of Golf3, we ask whether the cytosolic tail of this type 2 membrane protein, because Golgi enzyme are type 2 membrane protein, we were wondering whether the tail contained the information for Golgi retention. So to answer this question, we have taken this yellow protein, sucralose maltase, and what we have done is to swap the tail. We actually remove the tail of sucralose maltase and implant the tail of all the Golf3 clients and see where sucralose maltase localize. As you can see here in this immunofluorescence experiment, while sucralose maltase wild type localize on the membrane, all the chimera containing the tails of the Golf3 clients localize at the Golgi. So when you remove the adapter Golf3, then the Golgi localization is lost and the enzyme, the chimera move back to the plasma membrane. This suggests that the tails of the enzyme contain the information for Golgi retention, which is dependent by this adapter Golf3. And this is the first summary. So now we know that Golf3 binds and retains at the Golgi a set of glycosphingolipid biosynthetic enzyme by interacting with the motif in the cytosolic tails. Next, we decide to follow the trafficking of the main interactor of Golf3, that is lactosylceramide synthase. A synchronizable version in collaboration with Gaël Boncompagnie and Franck Perez generated this rush system. Using the rush system, we generate this lactosylceramide synthase, which can be accumulated in the ER and released in a specific manner. So what we have done is to follow the trafficking of lactosylceramide synthase in Golf3 knockdown cells and Golf3 overexpressing cells. And what we have found is that in the absence of the adapter Golf3, the enzyme cannot be retained anymore in the Golgi. So it localizes to this spot that I can anticipate to all of you, they are lysosome. While when Golf3 is overexpressed, that is actually what happens in cancer cell, the enzyme is retained more in the Golgi. Actually, the flux of the enzyme to the lysosome, it's impeded. Here, the immunofluorescence showing that this spot, as I was saying to you, they are lysosomes. And in other words, Golf3 controlling the localization of this Golgi enzyme affect their amount, meaning that Golf3 positively control the amount of this enzyme. Okay, when it's overexpressed, it's retained, and so the enzyme increases. When Golf3 is not there, the enzyme is lost. So we have done also electron microscopy to see what happened at the ultrastructural level. And for more simplicity, you can see in the center where there is a cartoon. So actually, lactosylceramide synthase is a trans-Golgi enzyme localized on the trans site of the Golgi and also appear in COP1 vesicles. When you overexpress Golf3, the enzyme increase in amount and also start to move back in the cis-medial site of the Golgi and also increase in this retrograde COP1 vesicles, while in the absence of Golf3, the Golgi is almost empty because the adapter is not there and the enzyme cannot be retained. But just to summarize this part, we have Golf3, which works as a rheostat, thus controlling the flux of his own clients from the Golgi to the lysosome. So when Golf3 is not there, the enzyme is not retained, move to this degradative compartment, and then it's lost. While when it's overexpressed, then the flux of the enzyme to the lysosome is impeded, and the enzyme, because of fast recycling, will increase in amount. Okay? Now we know that Golgi control the protein level and localization of sphingolipid glycosylating enzyme at the Golgi. And as I was saying before, these enzymes, they are part of the glycosphingolipid biosynthetic pathway. It's a complicated pathway. So for matter of time, I will just tell you that these glycosphingolipid are plasma membrane component. They are seated on the plasma membrane where they work as a receptor for virus, bacteria, but they are also component of the glycosphingolipid enriched microdomains. So they interact also with RTKs, thus modulating signaling. So the key enzyme regulated by Golf3 is that one indicated by the green arrowhead, lactosylceramide synthase. Two other things that I want to highlight about this pathway is that the ceramide, that is the precursor of glycosphingolipid, is a toxin compound, meaning it causes, when it's accumulated, causes top of cell growth and apoptosis, while the glycosylation of ceramide into complex glycosphingolipid that are globo, ganglios, yellow, or lactoside, they are usually pro-growth metabolites. Let's see what happens when you modulate Golf3. To check the pathway, I just want to tell you that we use several techniques. We have used sphingosine pulse chase experiments. So we give radioactive sphingosine and follow the production of the glycosphingolipid. It's the precursor sphingosine. Then we use mass spec. We use as a tool also toxins, which bind specific glycosphingolipid. These toxins were fluorescent, so we can follow by confocal microscopy or by FACS. Of all this technology, we find out that when you remove Golf3, the enzyme lost the Golgi localization, goes to the lysosome and is degraded. So the loss of lactosylceramide synthase will cause the increase of ceramide, while when Golgi is overexpressed, the enzyme is not lost, so and this will foster the consumption of ceramide glycosphingolipid. So since ceramide, the oncosuppressor molecule ceramide is decreased, this corresponds to an increase in complex glycosphingolipid, mainly GB3. So now we know that Golf3 controls sphingolipid glycosylation, but as I was telling you before, Golf3 is also an oncogene. It's an oncogene because when it's amplified, it's overexpressed, and this will cause the activation of mTOR, which in turn cause growth and proliferation. So we obviously ask whether sphingolipids are involved in this process. And by using, different technique like biochemical approaches, as well as growth assay and knockout cell line for lactosylceramide synthase, or the enzyme before glucosylceramide synthase. By the combination of all this assay, we find out that Golf3, here on the left side, via lactosylceramide synthase, control the glycosphingolipid production, which in turn activate mTOR, and by doing so, proliferation and tumorigenesis. So these are even more important if you look at the, pathology, meaning that what we have done is to take more than 70 patients with lung cancer, and we find that wherever you have a lot of Golf3 because of genome amplification or simply overexpression, then we also have a lot of lactosylceramide synthase. So increase of this enzyme because of the reason I just told you before. Okay, so this is an important data because this suggests that for Golf3 positive tumor, in theory, you can give glycosphingolipid inhibitors. Okay. Now we know that Golf3 promote mTOR signaling, impacting on glycosphingolipid levels. And I'm at the end. This is the working model. So when you have a tumor because of Golf3, what happens is that this adapter increases the cycling of this enzyme, thus preventing the loss of enzyme to the lysosome. This fast recycling will cause an increase of this enzyme, which will foster the glycosphingolipid production. So glycosphingolipid increase on the plasma membrane, and in a ligand-independent manner, they will activate signaling of the plasma membrane. So they activate mTOR, which in turn will cause uncontrolled cell proliferation and cancer. And this is most important slide. Here you can see all the people that participate to this work, all the institute, founding, obviously the Eurobioimaging, and all of you for your attention.